Hello, my name is Brio Hamal and I'm a student in Genetics 311. Today I'll be going over my Exam 4 mini video. I chose Prompt 2, so for my Exam 4 mini video, I'll be explaining transcription in prokaryotes. This is the transcription unit I created. The direction of transcription is going this way. We can start off by labeling our template strand. The template strand is complementary to RNA and is where the section of RNA will closely bind. With that being the template strand, we know that the top strand is the coding strand. The coding strand base sequence corresponds to the base sequence of the RNA transcript. As we continue along, we get to the promoter region. We have the -35 region and the -10 box. These are both important consensus sequences involved in RNA polymerase binding of a transcript. They get RNA polymerase to bind, which is very important. Also, within the -10 box is the PRIBNOW box. As we continue along, we get to the +1 site. This is the transcription start site, the start of transcription. This is where the first nucleotide is transcribed. As we continue, we have a large region here, which is the RNA coding region. As you go along, you get to the terminator. The terminator stops transcription by inverted repeats. RNA binds together and creates hairpin loops. Within these hairpin loops, a collection of uracils, binding to DNA allows RNA to release from RNA polymerase to terminate transcription. When transcription is terminated, the RNA goes to the ribosomes. A question asked was, if the -10 box is deleted in a gene, then what occurs? As we said before, the -35 box and the -10 box are very important in RNA polymerase binding. So if the -10 box was deleted, then the promoter recognition by RNA polymerase will not occur. Binding will not occur. As a result, transcription will not occur. And this can have very severe consequences on an organism. So the -10 box is very, very important to the transcription unit. I appreciate the time, and thank you so much. Have a great day.